Hello friends, I will discuss about the protein structure at its secondary structural aspect. I will talk about the backbone structure, different types of secondary structure, the bonds involved to impart protein secondary structure or protein stability, protein denaturation, and methods to determine the conformation of secondary structure of protein. Let's start. So, three-dimensional structure of the protein which is biologically active is made by different covalent and non-covalent interaction like van der Waals interaction, hydrophobic interaction, ion pair interaction, etc. So, that will actually make its primary as well as the secondary structure. In case of primary structure, only covalent peptide bond is there, but in case of secondary structure, so many different structures, so many different bonds are involved. So, secondary structure is local regional folding or local backbone structure made by intra or interchain hydrogen bonding. There are so many bonds like non-covalent, van der Waals, hydrophobic interactions, so many interactions are involved here to make the secondary structure. In 1951, Linus Pauling have proposed the secondary structure alpha helix and beta sheet model. Characteristics. Let's talk about the characteristics of the secondary structure. So, secondary structure is the local structure, the regional local backbone structure made by intra or interchain hydrogen bondings. Many R groups of amino acid chains are also involved here, and we can find different types of assemblies are present here and different types of non-covalent and covalent interactions are there. You cannot find always the ordered geometry of the secondary structure. There are some of the random and disordered structures are also present. Now there were some postulates or principle derived by Pauling by which actually the peptide bond structure was proposed. What were those postulates? He postulated some of the bond lengths and bond angles were distorted. Okay, number one. Number two, non-bonded atomic distance will not cross the van der Waals radii of the atoms itself. And in case of peptide bond, we know the C double bond and NH groups are there. They must be in the trans orientation. And there must be the presence of different types of non-covalent interactions are there. Based upon all those postulates, actually the peptide structure were characterized. So, we all know that peptide structure is made by, peptide bond is made by the dehydration reaction in between two neighboring amino acid residues. These peptide structures C double bond NH actually have some sort of electronic transition, CN bond. It is not the single bond in character. It has some double bond character. It is rigid and planar structure. Some delocalized electrons are getting transferred in between C and N. Okay. Now, the angle that is Cn in between carbon and nitrogen is 180 degree and omega, and it is in the same plane. Fine. But on either side of these plane, there are free rotations of different alpha carbon or amino acids. Now, all those free rotations are not too much flexible. They can rotate from 0 to plus 180 as well as 0 to minus 180. Now, what are those dihedral angles which actually give rise to the rotation? You can find the C alpha, right? This particular NC alpha angle is designated as phi, okay? And this COC alpha, this angle is designated as phi rotation. But all those rotations are not totally permissible depending upon the R group attached. Now, rotation flexibility can be determined by the Ramachandran plot. Now, here are the bonds and their corresponding angles involved to make the peptide structure. Now, what are they? COC alpha N is 110 degree, NCOO is 122 degree, CONH is 120 degree, C alpha CON is 117 degree, C alpha NH is 120 degree. The CC bond length is 1.54, Cn is 1.48 angstrom, and CO is 1.43 angstrom. The Cn, the peptide bond length, is actually 1.32, in between 1.49 single bond and double bond, that is 1.29. Okay, so it is in between them, that is 1.32, designated as it is not totally single bond, not totally double bond character. Here actually no eclipse structure are allowed, all are staggered structures. Ramachandran plot. Now allowed conformations, sterically allowed conformations of the tripeptide, depending upon the twist that it forms according to the formation of secondary structure. Some conformational maps are proposed because some structure are sterically forbidden, okay, but some structure are sterically allowed. The sterically allowed structure are conformationally mapped in two-dimensional graph or plot, that is called Ramachandran. That is called Ramachandran plot, which was proposed by G.N. Ramachandran. So you can find the phi and psi angles in different axes started from 0 to plus 180 and 0 to minus 180. The 75% of the plots are physically conformationally inaccessible, but there are several, mainly 3 points are there. Here you can find 4 points which are not sterically forbidden, and the secondary structures are plotted there. You can find the beta sheet allowed structure, the right-handed alpha helix structure, as well as some left-handed alpha helix structures are also allowed. And you can find some forbidden areas are near psi is equal to 0, very near about the allowed region. Okay, so some forbidden areas can be made to the allowed region by twists and all. So it is actually the Ramachandran plot. Factors. Now to make the secondary structure or helical structure, some factors are there like pH dependence and side chain R groups. We all know in case of polylysine at pH 12 only it makes the helical structure, but at pH 7 it cannot. Why? Because at pH 7 the lysine have all positive charge, so they will repel each other, but at pH 12 the neutralized structure help to make the twisted helical structure. The side chain R group, it is very important because when I will talk about the preference of amino acids, you can find in alpha helix some structures, some amino acids are allowed and some are not. That is also applicable for the beta sheets as well. We all know the proline having bulky structure, this is not allowed in case of beta sheets as well as in case of normal alpha helical structure, because this nitrogen is not actually involved to make the peptide bond, it is within the ring. So that actually creates the problem. Next, the prediction of secondary structure. The prediction by frequency P, actually the secondary structure is predicted according to the different amino acids, because some amino acids are allowed for alpha helix and some amino acids are allowed for the beta sheet as well as beta turn. In case of beta turn, aspartic acid is allowed, glutamic acid is allowed. In case of alpha helix, you can find the presence of alanine, methionine, etc. In case of beta sheet, what are the allowed amino acids? Mainly isoleucine, valine, they are allowed. Let's see what are the causes for occurrence of all those amino acids. Suppose in case of alpha helix there is a regular hydrogen bonding, fine. So if the serine threonine is present having hydroxyl group, that will tend to form extra hydrogen bonding, so interrupt helix structure. The bulky structure of the proline inhibit the helical geometry, as well as tiny structure of glycine also inhibit the helix structure. But you can find as well as isoleucine, valine, which are applicable for beta sheet structure, is not applicable for alpha helix. Why? It will impart more hydrophobic interaction, but that is allowed for beta sheet structure. But for beta sheet structure, glycine will not be able to make the hydrophobic interaction as well as alanine, so they are forbidden. Proline is allowed to make the hinge region in case of immunoglobulin, in case of helix structure, because after some continuous structure some hinge region can be present, but it is not yet allowed for beta sheet structure. Protein stability. To give the protein secondary structure stable, to stabilize them off, there are different types of bondings are there. What are they? Hydrogen bonding. In case of alpha helix, we can find intra-chain hydrogen bonding. In case of beta sheet, inter-chain hydrogen bonding is present, as well as the polar capping on either ends of the alpha helix. There is a polar capping made by differential hydrogen bondings, as well as hydrophobic interaction, CH2. So these actually impart the compactness of the protein because the hydrophobic residues are buried inside to avoid the thermodynamic interaction with the polar solvent. So polar structure, polar residues will protrude outside. Not only that, there are some feeble ion pair structure also, that is called as salt bridge interaction, made by NH3, extra NH3 group of lysine and extra COO minus group of aspartic or glutamic acid. That is called the salt bridge interaction. That is actually found to make the structure of hemoglobin. Apart from which, the disulfide linkages are very important, found in case of immunoglobulins as well. Disulfide structure make intra-chain or interchain bonding. As well as we can find various types of van der Waal interactions, that is permanent or induced dipole. If the dipoles are permanent, that is called, as the force is called Kizom force. If it is permanent which induces another dipole, that is called Debye force. If both the dipoles are made by the induction, that is called London dispersion force. But all other forces are involved to stabilize a protein structure. Apart from which, we know the Schiff base intermediate in between NH2 and aldehyde gave by the lysine in case of tropocollagen structure. Okay, so Schiff base can be made. We can find the secondary amide bond, that is also found in case of tropocollagen structure to bind the